Hello, good evening, good morning, everybody. I'm Dr. Asavari Manvikar. I would like to welcome you all to the CAR Global Webinar Research Series. Council for Ayurveda Research is an international non-profit organization based out of USA with a sister chapter in India by the name Ayurvidya Anusandhan Abhiyan Foundation, AAAF. Our mission is to encourage, educate, and facilitate basic clinical and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence-based health science globally. You can learn more about us at www.ayurvedaresearchusa.org. Please do consider joining us as a member, which will bring you access to all our resources. No nonprofit can operate and succeed without the volunteers. So consider joining us as a volunteer, and we can place you in a variety of projects based on your skills and your interest. Once again, our website is www.ayurvedaresearchusa.org. Now let me introduce our platinum sponsor, Komal Herbals. Komal Herbals is based out of Pennsylvania, USA. It uses all natural organic products in order to create various herbal products. If you want to know more about it, please go to www.komalherbals.com. Now, I would like to introduce you to our eminent speaker, Kiran Kini. She is MD, PhD in Ayurveda. She serves as a Director of Research. Namaste. She works as a professor in Roga Nidan at YMT Ayurvedic Medical College, Navi Mumbai. She has been approved as a faculty of HSET and RM since 2010, MUHS Nashik. She is an ex-member of CCRAS, Research Advisory Group, Ministry of Ayush, Government of India. She has published more than 23 research papers as a single author in double-blind, peer-reviewed research journals. She works as an MD and PhD guide at MUHS Nashik. She serves as a master trainer, CCIM, for HSET and RM by Central Council Ministry of Ayush. She has served as a principal investigator and co-principal investigator in many clinical trials and funded research projects. She has been invited as an honorable speaker on TISS affiliated All International Institutes for Leadership graduation program. She received Dhanvantari Puraskar for Sandhu Pharmacy Private Limited in 2022. She received Excellence Award by NEMA, India in 2019. She also received Thane Sevavrati Puraskar from Arogya Bharati and Arogya Dham in 2019. We are very fortunate to meet her today. She will be speaking about Ayurveda research from lab to clinic. I welcome you again. Now I would like you to take it from here.
Yes, thank you very much, Manvikar madam. I'm very much thankful to you for giving me an opportunity to share my thoughts on this eminent platform. So without wasting further time, let's start with our main topic, and that is Ayurveda research from lab to clinics. First, let me know, my slides are visible?
Yes, they are.
Yes, thank you so much. So may I start with?
Yes, absolutely.
Yeah. So today's topic is Ayurveda research from lab to clinics. Research is nowadays especially since last 4 to 5 years, research is very much demanding word in all over Ayurvedic fraternity, not only in India, but in foreign countries also, whether people are believe on Ayurveda practices. So, what is research? Sir Albert Einstein says, if we knew what we were doing, it would not be called as research, would it? Yes. Sir said, if we are calling something as research, then it must be. Hello, am I audible? Hello.
Yes, you are.
Audible.
Yes.
Yes.
Yeah. So Einstein sir says, if we knew what we were doing, it would not be called as research. So research is something that we are starting with some focus. And with that vision of that particular focus, we start planning about the procedure. And that planning should be systematic, it should be consistent, it should be scientific, it should be productive, and it should be focused. So objectives of today's session are, first, what is research and its types? What is Ayurveda research? Its need in the present era. Area of Ayurveda research. Ayurveda research in laboratory. And what is the pattern of Ayurveda research in clinic? And how this journey from lab to clinic in Ayurveda research. So in scientific language, one can say research is the systematic investigation into and study of materials and sources in order to establish facts and new conclusion. Whenever we think that we have to start for research, some planning or some question or some urge, very strong urge to do something new, which is very much important for society benefit point of view. That always used to be there in our mind, and we start searching what is the solution of the current problem. And for that search, we need always a systematic plan. That systematic plan with competent and authorized materials and related sources, so that we can establish the fact unbiasedly to form new conclusion. Whatever we assume for research, that is called as hypothesis. And to verify that particular hypothesis or that particular thing, we have to collect evidences or information from very scientific and authentic resources. So this is research. At the same time, research refers to a problem. Problem means when we observe there is some strong need of society, which or any problem in the society, we have to solve it for the benefits of mankind, for the benefit of the nature. What we do, we formulate the assumption. Assumptions means we think about that problem, and with the observation in the field, we form some solution, or we can say some hypothesis for that particular problem. And to prove that hypothesis on scientific base, we have to formulate a protocol. How we will proceed, how we can start with, how we have to go stepwise, what data we can collect, from where we can collect, what is the method of that, what is the exact center of that, and what are the observation criteria to analyze those facts which we have collected through the data. And that after in the last, we have to reach some certain conclusion in the form of solution, or to give some, to put some highlight on the solution of the concerned problem, okay, in certain manner. So it goes, this entire process is called as research, and it goes stepwise. This originates with a question or a problem. What we want to achieve, that we have to decide, or what we want to test, that we have to decide in the beginning of the research. And we should think very clearly. It requires a clear articulation of a goal. What kind of focus we are having in the beginning of the research, that should be very clear and that should be very specific. The entire plan should be very much precise, and we should divide the entire plan into two problems, that is principal problems and sub problems. Entire plan means your hypothesis. We have to divide that into the parts, and that is the principal problem and sub problem. Principal problem means specific research problem which we want to resolve or we want to get solution on that. So that is called as research, means that generates research questions. When we accept certain critical assumption, we start formulation of that particular process, and then it requires the systematic collection of the data. Data means scientific evidences that we can observe in context of that particular problem solution, which we are assuming as the hypothesis. And after this process, whatever data we will gather, we have to interpret that data with the help of statistics. And with the help of that interpretation, we should reach to discussion and conclusion. And this entire process is called as research process. So this research, types of research, the next topic is that these types of research, they based on two things. That is purpose, why we are doing that research. And second is methods, what we are going to achieve and how we are going to achieve. Okay. As well, these four main categories we could see in the practical field of the research. That is basic or fundamental research. Second is applied research. Third is evaluation research. And fourth is action research. Fundamental research enhances your knowledge about that particular point. It will not give the immediate economical growth to anyone, or you can say to the researcher, okay? But it will give the scientific view, or it will explore the scientific view behind that particular topic. Then second is applied research. Applied research is the problem solving research, where you are applying many of the, or you can say needed scientific theories and scientific conclusions, and you are trying to solve that problem. That is called as applied research. Then evaluation and action research, everyone knows. Evaluation is evaluating a process, evaluating certain plan, evaluating certain solution. That is evaluation research. And action research means you are doing something, you are giving some solution on that. So it is called as action research. And under action research, further types of research, they come. Okay. These are the other type of division, one can say, or this is the other picture of the research types which we use in Ayurveda frequently. That is called as applied research, basic research. These both things we have discussed in earlier slide. Then correlational research. What is the correlation between two factors? Descriptive research. Descriptive research means you have to describe certain things. Ethnographic research, experimental research, exploratory research. These three things are, many times you must have gone through these all types, because these three types are nowadays, last 5 to 6 years, we are frequently using this research in the context of Ayurveda research methodology. Then longitudinal is the research where you go longitudinally, same experiment in on same group, you repeat some observation. I'm trying my best to simplify the things in very simple words. Cross-sectional means at a time you are going to observe that particular event in different groups. Then historical research is the history about that particular scenario, or the history about science. Phenomenological research is the research about phenomena. Then two mainly important things, that is qualitative and quantitative research. They comes under after that. Qualitative deals with quality research, means qualitative or qualities of that particular group qualities of a particular thing, it is always a descriptive. We discuss about that, we observe about that. So it is called as qualitative. And then quantitative. Quantitative deals with numbers, empirical values. Okay. Then we are coming to Ayurveda research. When we are talking about Ayurveda research, what we have observed till date, there is a separate research methodology, guideline, or science in modern technology or in modern science. So there are some different guidelines for research in modern science, but in Ayurveda, we want different methodology than the modern, because these things are very much important in concern of Ayurveda research. That is subjectivity of the perceptions. Ayurveda deals with cognitives of Rogi and Vaidya. Rogi means to whom you are going to offer the treatment, and Vaidya means observer. So physician or observer, as depending on their cognitives, depending on their perception powers, or depending on their ability to understand the problem, understand the knowledge, understand the basic principles, we get different interpretations. So subjectivity is very much important in Ayurvedic research, and that we have to minimize. Or at certain level, we have to clarify to the researcher of Ayurveda that what we want to say about the particular term, and that is called as conversion of that subjectivity into objectivity. When you want to convert, or when you want to minimize that subjectivity, you have to set some standards of your observation things. What you will observe, how you will observe, what is the method of observation, what is the period of observation, when you have to observe. These all things should be similar everywhere when you want to do some research in the particular topic. So Ayurveda basic principles. Ayurveda has a lot of basic principles which we can read in every classical text or every classical Samhita of Ayurveda. And now also after 1,000 years of this all traditional knowledge, still these are very much applied, this universal truth by Ayurveda, still they are very much applied in the period of practices. So for this interpretation of basic skills, we have to adopt some theories, we have to adopt some practices, and we have to develop some interpretation skills. And those interpretation skills and practices that should be guided by Apta. Apta means those who are really enlightened with the depth of the knowledge of Ayurveda, those who have helped till date as an authentic source in the theoretical and skillful practices of the Ayurveda. We can call them Apta. So with the help of this Apta group, we have to enhance our skills of interpretation in practical point of view. Then we see the variety of Rogi Roga Pariksha, how to examine the patient, their methods and their types. Then Pariksha Bhav, how to observe, what to observe, and different tools of Ayurvedic examinations. These are the main important, or you can say main or important theories, or these are the main and powerful whole points of Ayurveda research, which makes Ayurveda research something special or something outstanding from the modern research team. So these all points should be studied and should be understood with the help of classical text as well as Aptas who have practices since long time successfully, who have delivered Ayurvedic results with appropriate and scientific interpretation to the patients, who is very much sensitive and ethical towards society. These all type of persons, they will help you to grow as a Ayurveda researcher. Then all our 14 subjects which are right now included in our syllabus. Basically, we say Ashtanga Ayurved, Kaya, Bala, Graha, Urdhvanga, Shalya, Dramsha, Jara, Vrusha. Apart from this, these all are Anga. We currently teach through 14 subjects by Ayush Ministries NCSM, As per the stipulated norm, there used to be 14 subjects for Ayurveda students in Ayurveda medical colleges, and those all covers all aspects or dimension of Ashtanga Ayurveda. So what is the need? What is the need of research in Ayurveda? We need standardization. We need standardization of the method, of the medicine, of the procedure, of the preparation, so that we can use this methodology, which is standardized all over in world, all over in not only India, all over in world. So we need standardization. So we have to develop some methods. For that standardization, we have to develop some methods, and work is going on. Everyone who is interested in Ayurveda knows what is the meaning of standardization. So standardization of treatment procedure. Then what are basic treatment guidelines? What medicine one has to, any Vaidya has to offer to patients, and what is the thought behind that process? How to think stepwise? What are the main markers or what are the main indications for choice of drugs? What are the main indication? What are the main guidelines for diagnosis of that particular situation or particular dosha dushya samurchana in specific patient? These all guidelines should be developed, and that should be presented, or that should be demonstrated on the base of science, recent science or life science, one can say. We have to emphasize literally work. We have to do many experiments, and we have to elaborate clinical research in every possible aspect. We have to revalidate whatever principles were told in Ayurveda classics, whether they are applicable and at what level, what are their markers, what are their frame. We have to check that. And for that, revalidation is the need of the current era. Then we have to find better treatment modalities for every situation, because Ayurveda respects autonomy. And in this condition, every need, so as per the Ayurveda's dhaya, swasthasya swasthya rakshanam, aturasya vikara prashamanancha, we have to see the better treatment modalities as per the need of the society. We have to establish dose, duration, indication, and side effects also. Every medicine which is having effects will give you some after effects also. Whether with the help of proper standardization procedure or proper preparation methods, we can enhance the properties, or we can enhance the actually the actual form of that particular medicine. And for that purpose, we have to be more focused for the standardization of each and every procedure. So this is the need. Now we will start talking about what are the areas of Ayurveda research. So fundamental principles about all things which I have told earlier in our classical grantha, that is Charak, Shushruta, Vagbhat, and Nidan. Whatever we refer as a classical text, all Upasanhita manuscripts, some fundamental principles are there that should be revalidated timely, and we have to start using that for our society in different contexts. For example, when we are searching the newer diagnostic tools, there also we have to say it as per Ayurveda, what kind of diagnostic tools we can develop through our different observation skills, which are very important for clinical practices. And that should be strictly based on fundamental principle. At the same time, this Kaya Chikitsa, Panchakarma, Shalya, Shalakya, Stri Prasuti, then all preclinical and paraclinical subject, that is Rachna, Sharira Kriya, Dravya Guna, Sanhita, Roga Nidana, Agadatantra, Swastha Vrutta, these all area needs research in Ayurveda for the betterment, for the better solution, for the precise practices, and for the standardization in this every area of Ayurvedic subject. We need to do research systematically with the help of Ayurvedic guidelines. So how we go in Ayurvedic research? How we have to start with Ayurvedic research? So first we have to decide the area of our interest, because research is very critical process. It is like very much, many times it is very exhausting, because what you are going to do under research, it must be very much new to everyone. As I said earlier, great sir, Dr. Einstein also said, if anything is known to you, it will be not called as research. So you need consistency, you need focus, you need dedication for that particular topic. So that area of that research, it must be your area of interest. Then you have to see what is the need of the society. We should not forget that we all are community helpers. So we are there to give the solution to the problems in the society. So first we have to see what are the problems in terms of, means any problem, psychological, physical, social, whatever. So we have to see what is the need of the society, and accordingly we have to see what is the gap in scientific knowledge and that need of the society. What we have to see regarding solution of that particular problem of the society, what kind of knowledge is established on scientific basis, and what is the need of current, or what is the solution of the current problem of the society. And to fulfill that knowledge gap, we have to do the planning stepwise. We have to see first what we want to achieve, that comes under aim. How we will like, what objectives we have to design to achieve the goals, that is called as objective. Then we have to see what are the materials and methods, what is the criteria that causes inclusion and exclusion criteria, where we can conduct that research, what is the sample size. Sample size will be dependent on occurrence of that particular problem in the society, and with the ratio or proportion of that occurrence, we can plan or calculate the sample size. And we have to decide what kind of observation we have to do as per the objective of our research project. So this all comes under the planning. When we are talking about execution, we have to go again stepwise. First, we have to do a chart or table. Whatever time is allotted for that research, we have to plan accordingly and stepwise, and we have to see how we can execute that entire process. Okay. Then we go to observational table. What kind of observations or results we want to record, and how it will be recorded. Everything will give you the outcome. And whatever outcome you will receive, you have to give proper discussion and conclusion about it. And you have to publish that particular research outcome. Unless and until you are not publishing that properly in scientific guidelines, your research will be incompleted. So this is the process, general process, and that's how we go in Ayurveda for research. So how actual the research is called, or how the actual research is expected in Ayurveda? Laboratory. It is always preclinical. Preclinical, everyone knows what is the word of preclinical. Whatever you do before your clinics. That is in vitro and in vivo. Means when you want to authentify, or when you want to standardize, or when you want to identify the new molecule or new drug, or any chemistry of that particular medicine, biochemistry of that particular effect, means biochemistry after that particular effect of that particular medicine, you have to go to laboratory, and in laboratory you have to perform many tests. Okay, so those tests can be done on animals, different kind of animals, or cell lines, or you can say in different, with the help of different methods of research. And you have to perform that preclinical research, means to prove new invented or some formulation which is said in the granthas but not still proven effectively and scientifically in practices. So this kind of things you have to go through in preclinical and after that clinical research. So we have to say what methods we are using, what drugs which we are going to use, the preparation and standardization of those drugs, the all raw material. It should be identification and authentication of that raw material should be done properly with the help of scientific guidelines. Then we have to see the efficacy of that particular pulp, and then we have to think about adverse drug reaction. Adverse drug reaction is very much important point for clinical practices. So when we are talking about drug research, biological activities in the forms of potential efficacy and safety, that is very important for drug research. If we want to suggest some proper dosage for the better patient compliance, we have to do the drug research. And if we want to demonstrate the long-term efficacy through clinical studies, then we have to follow all scientific guidelines of drug research. Okay, so in preclinical, these few techniques are very much important. As madam told me, this is very introductory lecture, so I'm trying my best to simplify the points and to elaborate what is the research, what are those types of that particular research which is demanding in Ayurveda. What is how Ayurveda research is different from the modern research? What are those areas? And for Ayurvedic research, what is the need? What are the areas? And how we have to develop or we have to think about the steps, that is preclinical and clinical and techniques. And still after that, these 2 or 3 slides, we will discuss some more topics. That is, what are the challenges and what are the limitations of Ayurveda research also. So now we are discussing techniques of physiochemical aspects of Ayurveda drug research. So as Ayurveda said, there is no substance in the world which cannot be used as a medicine. And Pinde Brahmande Nyaya, the next principle is whatever principles are present in the universe, they are also present in man. With the help of these 2 basic principles, we have to think about each and every substance in the world. This is which is very useful for Ayurvedic medicine. So Ayurvedic medicines are used to be very different types, means herbal medicine, herbo-mineral medicine, and there are many types of Ayurvedic medicine. For this, all groups, when we start thinking about the preparation of that particular medicine, we have to start with first physiochemical aspect of that particular drug. So the biochemical study of the fundamental units of life is the same. So it is very essential. And we have to see what are the effects of that particular drug on the cell, or in context of pH, buffers, as well as this electrophoretic techniques are there. These electrophoretic principles everyone knows because that is a part of basic education. Then chromatography, that is gas, liquid, and ion exchange chromatography. We use to identify the exact type of that, or you can say that exact type of pattern of that physics or chemistry of that particular drug molecule. So we use chromatography, electrophoretic techniques. Then we use different centrifuge machines, centrifugation technique. Then we go for tissue culture. Then we use radioimmunoassay. Then we use enzyme immunoassay, that is ELISA. Then we use spectrophotometry, electron spin resonance spectrometry, biosensors, and synthetic chemistry. These are basic things which are important to know the physics and chemistry aspect, physico-chemical aspect of Ayurveda drug. These all tests should be performed in laboratory with the help of different scientific techniques. Okay. Then the we will discuss regarding after physicochemical, we have to discuss with biological methods of drug research. What are the biological methods? Importantly and frequently used are animal models. So they will give us the limitations, efficacy, idea about dose and biological action of that particular drug on animal cells. So different types of cells we require: respiratory tract cell, intestinal cell, and different cell lines. Of course, cells means cell lines. Okay. So in vivo, when we see different animals we use, that is cat, dogs, rabbits, guinea pigs, rats, and mice also we use. And for oral or injection, we use subcutaneous or intraperitoneal, intramuscular, and intravenous injection, or we give the oral feed to these all animals. Then we form some experimental models on of some diseases. Means we induce some diseasal condition in that animal, and we see what are the effects of invented drugs or invented Ayurveda formulation on that particular animal or that particular cell line. Then we have to see the animal toxicology profile after introduction of that particular drug into that animal. We have to see what is the toxicological profile. Then symptoms recorded during the acute toxicity studies. Then functional test, which we use in experimental toxicological study. So this is all about Ayurveda preclinical for Ayurvedic drug development, that is physiochemical, and after that biological test. Then we come into Ayurveda research in clinics. Okay, so clinics, what are the basic things which are required by Ayurveda point of view? That first is Rogi Roga Pariksha. So first we have to learn what is the scientific interpretation of classical Rogi Roga Pariksha methods, and we have to see how we can convert the maximum subjectivity into objectivity. And for that purpose, we have to develop some parameters. We have to standardize some protocols so that Rogi and Roga Pariksha methods will be similar everywhere in every Ayurveda clinic. Then we have to see what are the interventions. Intervention type, intervention periods, for that intervention doses, methods that should be standardized. Then we are thinking about adverse drug reaction, which is unexpected or undue towards the expected result. And this is very much harmful for physical or mental health or status of that patient. Everything has to be recorded very well. If you expect XYZ result in that particular patient and you get ABC, then also you have to record that everything and you have to report as per the protocol or as per the method wise, scientific method wise. Okay. Teaching and learning methods and its effect. With means in clinics also, whatever we are teaching to our students or we are teaching in our institute, we need to upgrade those teaching and learning methods, as there is the means everyone can observe in their home also. This next generation, this generation is very fast. They are very fast learners, but they don't have a lot of patience. So considering these all factors, if we want to pass the authentic Ayurveda in the next generation of Ayurveda practitioners, then we have to change our teaching and learning methods according to the need of the time. Okay, so we have to invent newer and very much practical teaching and learning methods, and we have to record their effects. That is also a part of Ayurveda research. Okay, then the next is methods of administration of medicine. How we have to take that medicine, how we have to, means there are certain scales where are told in Ayurveda basic principles regarding administration of medicines, how they're effective. If they will be not followed, what kind of things are observed, can be observed. And the observation should have some scientific parameters also. So that again, we have to develop. We Ayurvedic practitioners, we carry a lot of responsibility more than laboratory scientists, because actual clinics are the main origin of newer topics of Ayurveda research. Then any Ayurveda clinician practices Ayurvedic philosophy, or you can say Ayurveda principles for their patients, they may observe, or they have to, or they must observe so many things, because Ayurveda always believe that everyone has to develop their own skill, and every practitioner should respect the autonomy of every patient. Every patient is different from each other, and all basic principles of Ayurveda indicates very clearly that every case is different from each other. So in the special context of every new patient, we have to observe many things, and that observation method should be also standardized. Okay, so this is again the new area of Ayurveda research. Then Panchakarma and Kriya Kalpa, different experiments we need to do in the patients with the help of basic principles of Panchakarma, of Kriya Kalpa. Many innovations are expected as per the need of that patient. And again, this is the area of Ayurveda research. If you observe new innovations, which is very effective in Ayurveda practices, you have to record them very carefully, and you have to test that innovation or that particular upgradation in larger scale of Ayurvedic patients. And you have to plan accordingly how you can observe that particular thing in the larger scale, and what are the parameters. And for this purpose, you need some systematic plan, and that is called as Ayurvedic research methodology. Then how you will select the topic? First, you have to just discuss the same way. We have to see the area of interest, and accordingly, you have to identify our topic. This is the selecting topic is the first step. Second is you have to refine your topic. You will be probably have to narrow your topic, since most topics are too broad for a research paper. So you have to be very clear after doing a lot of paperwork. You have to be very precise what kind of work you want to do, which kind of research you want to do. If you are doing some applied research, then also what exactly kind of applied research you want to do. And you have to develop your topic with the help of your guide or experts, or you can say those who are practicing Ayurveda since very long time. Accordingly, you have to develop your topic. And then once you have identified your topic, you have to see what is the strategy.
Madam, please unmute yourself. You are muted.
I'm muted.
Now we can hear you.
This is now okay.
We cannot hear you.
Hello. Now it is
Yes.
better.
Now this is good.
Yes. Yes. So I was talking about choosing the research topic. So according to when once you have identified your topic, you have to form the strategy, how you are going to search that particular outcome on the basis of science. So if that topic or that subject is your interest subject, then you have to first completely have to be very much immersed into journal articles. You have to read more and more scientific data, and you have to concentrate on a single research question, and try to put the effort to be adaptable. You have to set your mindset in such a way that you have to read more, you have to change yourself, and you should have very much flexible mind to accept every new observation which is required for that particular topic. At the same time, you have to update yourself for the latest advances. You have to keep learning, reading in your industry, and you have to discuss more and more with different scientific thought processes. You have to talk with your friends about your research idea. When you want to do the thorough research in Ayurveda, you have to be always original. You have to be inventive, and you have to have very much specific interest which suits to your practices, because ultimately this research you are going to use in your practices. Okay. So these are the 7 steps of research process. After getting developed the idea, or if at the point where you are very much clear with your idea, what you want to do, you have to start this research process into 7 steps that find the first is identifying, second is finding background information, what is proven, created on scientific basis. Then you have to use catalogs to find books and media. You have to see the databases to find journal articles. You have to see the internet, and then you have to search what you have found, and you have to very much selective about the standard format which you can use. And again, in your methodology, you should be able to give the answers of this 5 questions, 5 WH questions. That is why you are choosing that topic. What is the source? Who is the source of information? What is the most important question? Where your topic matters, and when your topic is like they will give the solution, is or was your topic is important, and what is the current issue, and your how much your topic is relating with the current issue. Okay. Then the most crucial part is keeping your motivated during this journey, and the supervision is very much important. It is also critical part, and supervision should be done to keep you going. And as a result of this, a good and effective topic is one that can be given to the compelling arguments that are relevant to the current practices. And as far as possible, you have to be very much ready for brainstorming regarding the subject matter. Think about your expertise and interest. We have to do a lot of literature review. Okay. We have to think, keep in watch about our feasibility. So that topic is very much attractive, but economically, socially, time-wise, is it feasible for you? That you have to think twice. And you have to be very specific, and you have to be open for feedback. Okay. So these are the things where how we can start the Ayurveda research. But what is to be avoided? First, played out topics, then personal stories, personal interest and information, then the topics which don't have any available scientific information, which is too technical, too narrow, too broad, or too theoretical. It is not at all significant or controversial, that we have to avoid. And these are few things which we have to keep in mind. Why many times researchers are getting failed or getting stuck. So first is the lack of scientific training, how to go for research methodology, how to go for the methodology of the research. That is very important. Then it is very insufficiency regarding the interaction between the different sources of research, either they are both in universities or business establishments or government departments. Lack of communication, then materials quality, then overlapping studies. Okay. Because there is no any interaction or adequate information about the completed research. Then many times code of conduct, many people or researchers, they miss that, so their research gets failed. Interdepartmental activities or rivalries, these are the one of the main reasons every one of you can understand. And assistance is always important assistance, including computational assistance, your knowledge, your skills, library management, functioning. You have to read more and more. You have to spend tracing the books, journals, reports. Keywords are not the enough thing. You have to read all around in 360 degree about your topic, and you have to proceed for your further research. That's all from my side. Thank you very much, because 35 minutes were given to me, and this was very introductory lecture. So I've tried my best to cover everything. So that's all from my side. And now this session is open for discussion or questions. Thank you very much.
Thank you very much. This was amazing. Very informative, very stepwise, very organized lecture. It will be actually very, very useful for all the MD students as well as PhD students. In the US, we have Ayurvedic Health Counselor, Ayurvedic Practitioner, and Ayurvedic Doctor programs. and they are also supposed to do a research, and they are supposed to write a research paper. So many times the students are not aware exactly how to choose a topic, what is what is expected for them to do. So this lecture will be really very good for them to start with. Again, thank you so much, Dr. Kini.
Thank you. Thank you very much, madam, for giving me an opportunity on this eminent platform. Research is such a vast topic, and nowadays everyone is having the person or the scholar, Ayurveda research who want Ayurveda student who want to do the research. They are like thinking, what is the exact way to do? What is the exact methodology which we can apply for Ayurveda research? Because general modern research guidelines are not always applicable for Ayurveda research. The person who is doing good Ayurveda practices in the field, authentic and ethical practices, they can tell you regarding this. But we should not overlook, or we should not miss the basic points of Ayurveda research, how we have to go from preclinical to clinical. What are the steps? What are the areas of preclinical, and what are the mandatory steps in preclinical? And when we are talking about clinical, what things should be done as per minimum standards in clinical practices? That should be very much taught by different through different workshops and seminars. And this all entire training is preclinical training, thought process training, interpretation training, designing training is very much important for every Ayurveda researcher. And that is my opinion. Once again, I'm thanking you for this golden opportunity. And that's all from my side.
Thank you. Thank you so much. So I will be uploading this recording to our CAR YouTube channel. Please go to our YouTube channel. Everyone who was able to attend today's lecture, as well as everyone who is going to, who is interested into research topics, learning about various Ayurveda research that is going on in different areas, please go to our YouTube channel and definitely like, share, as well as subscribe our YouTube channel, so that you will be able to learn about previous topics as well as different lectures and lectures which we will be conducting in the future. I see Dr. Rajan Patankar here. Sir, do you have a question?
No questions, but very excellent, precise, and step by step. As she is very well known in this field, how to present each and everything. So thanks a lot for giving our opportunity, and thank you. Thank you very much.
Thank you. Thank you, everyone. Namaste. Hope to see you next time.